stay a while and listen.
The impact of plant polyphenol on ovarian aging. Many underlying molecular mechanisms are at work. November 19, 2025. Share. Plant polyphenol, such as resveratrol and quercetin, have been documented to have beneficial effects against ovarian aging in various experiments. Some human clinical trials have reported related benefits. These benefits are attributed to anti-inflammatory activities, hormonal effects, the reduction of oxidative stress, and effects on gut microbiota. A recent review in the Journal of Ovarian Research summarizes current knowledge of the impact of various polyphenol on different aspects of ovarian aging. The researchers discuss that polyphenol supplementation could be used as an intervention to delay ovarian aging. 1. Every woman's problem. Ovarian aging and cessation of proper ovarian functioning precede aging in other organs. Ovarian aging is related to a reduction of oocyte quality and quantity, which is the main reason for age-related infertility. Beyond the fertility problems, ovarian aging impacts lifespan and is also linked to many age-related conditions, such as osteoporosis, cardiovascular disease, and neurodegenerative disorders. Therefore, finding ways to delay it is in the interest of every female, regardless of her childbearing goals. At this moment, hormone replacement therapy and assisted reproductive technologies are used to address ovarian aging-related problems. However, they are unable to reverse female reproductive aging and the declining ovarian reserve. Longevity researchers are currently seeking ways to extend the female reproductive span, but before effective therapies are on the market, lifestyle factors can be utilized to address ovarian aging. The authors of this review highlight polyphenol, naturally occurring metabolites found in fruits, vegetables, nuts, seeds, herbs, spices, and medicinal plants, as one possible intervention. Polyphenol exhibits strong biological activities, including antioxidant, anti-inflammatory, antibacterial, and antiviral properties, and demonstrate numerous beneficial effects. Studies also suggest that they can reduce the risk of cardiovascular disease and neurodegenerative disorders, too. Delaying ovarian aging. The first part of this review discusses resveratrol, a plant-derived polyphenol that has antioxidant and anti-apoptotic properties. Multiple studies have shown that resveratrol's dietary supplementation or oral administration alleviated ovarian oxidative stress damage, restored hormone levels, reduced ovarian cell apoptosis, and improved reproductive performance in animals 3 to 7. It also positively affected epigenetic changes and gene expression in the aging ovary. Polyphenol extracted from tea leaves were shown to reduce inflammatory responses and oxidative stress and to improve ovarian reserve and ovarian function in animal models of induced ovarian damage. 8, 9. Human, mice, and other mammalian research also suggested their benefits in alleviating ovarian aging and improving reproductive performance by enhancing oocyte quality and reducing oxidative stress. 10 to 12. Quercetin was described as having strong antioxidant properties. It can promote in vitro maturation of oocytes from aged mice and humans. 13, and slow down the aging of human ovarian cells. 14. Experiments in middle-aged mice also showed that oral administration improved estrus cycles, pregnancy rate, and ovarian reserve. 14. In polycystic ovary syndrome, PCOS, models, quercetin reversed many detrimental changes, such as irregular ovulation and hormone secretion, or increased ovarian cell apoptosis and inflammation. 15, 16. Similar beneficial effects were also seen in experiments in livestock and poultry animals. Proanthocyanidins are among the most potent natural antioxidants and possess several other beneficial characteristics. They have been linked to ovarian health in multiple studies. Research in rodents and human cells reported that proanthocyanidins reduced oxidative stress, inhibited ovarian cells apoptosis, improved oocyte viability and quality, modulated hormone levels, and alleviated PCOS. 17 to 20. Less commonly studied polyphenol also mitigate ovarian aging. Curcumin has been found to delay the ovarian aging process by increasing follicular number, modulating hormone secretion, reducing oxidative stress, enhancing oocyte maturation and embryo development in an aged mouse model. 21. Other polyphenol, such as chlorogenic acid, ferulic acid, and pterostilbene, have been shown to positively impact ovarian reserve, ovarian function, and oocyte quality by mitigating oxidative stress, reducing ovarian cell apoptosis, and reducing DNA damage. 22 to 25. Advertisement. It is worth noting that several studies have shown that a moderate dose of polyphenol can make a profound difference, and high doses of polyphenol can be toxic and cause ovarian damage and impair oocyte maturation. 17, 26 to 29. Many mechanisms, one goal. The beneficial effects of polyphenol can be achieved through modulations of many molecular pathways. One of them involves oxidative stress and the excessive production of reactive oxygen species, ROS, which can contribute to an increase in inflammation and disrupt the redox balance, leading to damage to mitochondrial function, telomere shortening, apoptosis, and inflammation, all of which compromise the proper functioning of ovaries and contribute to ovarian aging. Since polyphenol have antioxidant properties, the researchers investigated their role in delaying ovarian aging by examining the effects of resveratrol, quercetin, and epigallocatechin gallate, EGCG. Those polyphenol have been shown to modulate multiple molecular signaling pathways related to oxidative stress, improve ovarian antioxidant capacity, and reduce ovarian cell apoptosis. 30 to 32. A human randomized controlled trial also showed a positive role of plant polyphenol, curcumin or resveratrol supplementation, in alleviating ovarian aging by reducing oxidative stress. 33, 34. Polyphenol anti-inflammatory properties can also help alleviate ovarian aging by reducing inflammation, which negatively impacts ovarian health. These benefits were demonstrated in animal and human models of ovarian damage. 835. Reduction of inflammation and improved oocyte and embryo quality resulted from polyphenol supplementation in women with PCOS who received quercetin. 36. However, there is still a need for research on the impact of polyphenol on inflammation markers in naturally aging ovaries. Aging-related hormonal dysregulation significantly impacts ovarian aging. The hypothalamic pituitary ovary, HPO, axis controls hormones related to the reproductive system. Aging-related dysregulation of the HPO axis contributes to the depletion of the ovarian reserve and decreased quality and quantity of ovarian cells. 37. A growing body of research suggests that plant polyphenol regulate the sensitivity and secretion of reproductive hormones, potentially mitigating ovarian aging. The impact of polyphenol on hormonal balance and ovarian health was tested in preclinical and clinical studies of women with PCOS, showing their benefits for ovarian health through the regulation of hormone secretion. 38 to 40. The microenvironment surrounding the ovaries is also essential. This environment normally supports ovary and oocyte maturation, but aging leads to the accumulation of metabolites that might disrupt the homeostasis. The gut microbiota and its metabolites influence the ovarian microenvironment via the gut-ovary axis. Through this axis, the ovary communicates with the gut microbiota via hormone secretion. On the other hand, gut microbes produce metabolic signaling molecules that can impact ovarian function. Studies that used fecal microbiota transplants suggest that maintaining a youthful gut microbiota may help preserve ovarian function and reproductive health. 41. Similarly, experiments with laying hens as a model suggested that polyphenol supplementation could improve ovarian function by modulating the gut microbiota. 27, 42. There appear to be many mechanisms and pathways through which polyphenol impact ovarian aging. This is unsurprising since polyphenol are a broad group of molecules with diverse chemical structures, resulting in distinct bioactivity profiles. This knowledge can be used to design safe plant polyphenol-based interventions for female reproductive longevity that can be used alone or in combination with other treatments.